I'm gonna be talking today about a topic that is very dear to my heart, and it has to do with starting and moving projects forward, whether you're in academia or in business. But there's still commonalities in terms of strategy and end goals that apply to to both types of environments. And I'm not gonna go over my background. Marcus did a fabulous job. Um, but yeah, the MBA, I have to say, was one of the things that gave me even more insight in terms of incorporating a business approach to doing research. So for today, I'm gonna be talking about primarily 5 points. The one, it's gonna be about starting a research program, and then how you go ahead and find that good molecule that you need eventually to get things off the ground, moving it along the way, in a way that you can advance it quickly without breaking the bank. And a point that I think a lot of you might find very interesting is about where you can get the money to do all of this. And at the end, I would like to wrap it up just with some skills that might be useful to you in life, not only inside of the lab but also outside of the lab. So we need to start at research at from some point. Typically, it's biology. So when it comes down then to biology, it's all about the biological target. And when it comes down then to the biological target, we know we can have a biological target that might be known already. You just happen to be interested for whatever reason that you have scientifically. But at the same time, it could be a new biological target. Maybe your lab just discovered it, or a partner that you're working with. Another point that we need to know when it comes down to biological target is what it does and how it works. What is it that it does, and what kind of impact it has. Since we're talking about drug discovery and pharmaceutical drug development, how is that tied to a particular disease? And we need to have tools to monitor and know what it does, what the biological target does and how we influence its role. For that, you have to have assays. And from my point, which is structural-based drug design, knowing the structure, structural info on the target is biological target, helps a lot. So that's on a high level, knowing about the biological target. Now on the chemistry side, we can have compounds. Maybe you have active compounds against a biological target. So some compounds may be known, and based on that, you can assess what kind of compounds that you have in hand. Can be peptides, peptoids, could be nucleotides, could be sugar, small molecules, natural products, and other kinds. Eventually, we need to know how is it that they work, and that helps a lot. But in many cases, we can have a molecule that is active, but we have no clue as to how it does what it does. That's typically a case when you do phenotypic assays, where when you test a compound against, let's say, a cell, you see something, a phenotype effect, but there's no idea how it is working. And lastly, you have to either at that point have a molecule that you already have and you just want to start moving things forward, or in many cases you just need to find one to get you started from a chemistry perspective. So knowing what I call the space is important. And when it comes down to a biological target, we're talking about the biological space. And 5 points that we can discuss is critical factors for knowing the space is the function of the biological target, how it is that it works, but put it in a context as to not only what it does, how it works, but where is it found in the cell, where it's expressed either in the body or in the cell, how it is activated, and what happens after it does its function. So all of that starts giving you a map of everything that goes around the biological target that you need to know. That helps you figure out, okay, what kind of biological assets then you have to have at hand, because if you are mapping, everything that goes around the biological target, then somehow you have to monitor, those functions. And I'm included here similars because in many cases we're in a situation where the biological target that we're working on it's not unique. I mean, there might be other targets, other biological entities that are highly homologous to our target of interest, and that means that when we start drugging, our target, we might be hitting other targets, and you need to know that and figure out how to monitor that. Eventually, when things have to happen, well, you need to have access to that biological entity. So where can you find it? Is it something that it can be found, in commercial places, or somebody in your lab or outside of your lab, might have to get it for you. So you need to know how it is that you can access it, so you can, run your biological assays with it. And you need to know everything that goes on, around that. And for that, we all know that we can go to scientific publications to learn as much as we can about the biological target. And patents is also another source of information. Sometimes might not be as detailed as a scientific publication, but still, typically patents are the first ones that come and tell you something that eventually is published in more detail. And ultimately, who's working in your area, what I call the players. If you read publications, if you read patents, you can tell, who are the authors, and there's association, who, are they in industry or in particular university. That tells you, who the players are in that field, and you can use that for awareness. This is a very simplified snapshot of what might happen in certain part of the cell from a signaling pathway. And in this case, again, it can be more complicated in real life. This is just a quick example where we can see that if working on a particular target may have a lot of roots that touch it before and after. So part of what I call the biological space here. Now, on the chemistry side, we need, from a chemistry perspective, if we happen to have a variety of compounds at our disposal, now we need to look into the variety of them. What kind of compounds we're talking about, what kind of scaffolds we have in front of us, and assess their type and complexity. That has implications when we start making decisions as to what to consider and how to prioritize. And we're going to be talking about that a little bit, later in the presentation. In the same way, we want to know how they influence the biological target, what the mode of action is, and the mode of action can be in different ways. Sometimes can be known, sometimes can be new. But knowing how they influence the biological target, that's critical. At the same time, the chemical structures can tell you potential liabilities, not only from a chemical perspective in terms of stability, but also druggability. If you have highly reactive compounds, you don't want those. We're going to be talking about that a little bit later. And drug liabilities, something that might have problems once that is in vivo system, they metabolize very quickly or excrete very quickly. Many things that can come and play a role in the role of the compound in an in vivo system. And just as I mentioned, for the biological space, same thing. Publications and patents are going to give us, the information that we need to start knowing not only what kind of chemicals and structures are in a particular space, but also who's working on those and who the players are. So now we're going to start looking into setting our strategy when it comes to looking at the chemistry space, for instance. Here, I failed to mention, this is, I'm giving an example compounds that are involved in PI3K inhibition as an example. And this is not comprehensive, just about 19, 20 examples, but there are more out there. I just didn't want to crowd the screen. But when we look at this, we're going to start looking at the chemical diversity of the compounds. And in that case, it's going to be the type of compounds that we have with us. It can be that this is maybe a natural product. Maybe the other ones are synthetic. We can have some of them can be very simple. Others can be a little larger in terms of molecular shape. Others can be a little bit more complex, like when you start seeing chiral centers as part of the molecular structure. So that's something that we start looking into that. At the same time, we can see how crowded or how empty the space is from a chemistry perspective. If we have 50, 100 different type of scaffolds, that is telling us that it can be crowded. It might be a little difficult to enter into that space. Or we may find that in an example that we only have 3 or 4 chemical entities known. So that might be opportunity there for a research program. And that starts giving us an idea also about where we consider chemistry holes, things that are at this point are not present, that with your intuition and expertise, you must start thinking, oh, here's something that has not been published at this point in time. So we start looking into what kind of chemicals we can use for our research, and pretty much we have 2 ways. One is we can come and try to design a compound. One strategy that is commonly used is, for instance, you look at the structure of a compound and you start making variations of it to see if such a chemical entity has been ever reported. In this case, for instance, we can look into see if a similar analog where the oxygen has been changed to a nitrogen or a sulfur has been reported, or if one of the carbons here in this phenyl ring has been changed to a nitrogen, for instance, or in this case, the nitrogens of the 5-member ring, they have been moved around, and although they regioisomers, they might still be new entities that might offer opportunities there. The other approach is when we go and get compounds that are commercially available, and you just want to go and find, something that might be of use for a particular target. And in this case, eventually, through the computational modeling, we need to test those compounds, which is going to validate the biology. And if the biology is what we were expecting to see, like let's say inhibition of the target, then that starts validating the model. So we start looking now at the biological target. We need to create a 3D structure of it, especially if we're looking at this from a structure-based drug design approach. And getting the structure of the target is primarily obtained either by NMR data or X-ray data. There's a lot of there's NMR data and X-ray data at the protein data bank, and each one has pros and cons. NMR data, it's very good information because we have that the target is in solution, which means that we're looking at a system that is very likely is going to exist in the body or in the cell. Flexibility is part of it, but obtaining that type of structural information takes a lot of time. And if you go to the protein data bank, you're going to find structural NMR data, but it's rather limited. There's compared to crystal X-rays data. So the X-ray data is more available, as I just said, and it's easier to solve a crystal. I'm not saying that it's easy. I'm just saying that it's easier. The reason is now we have automation that allows you to change solving conditions and many other things, very quickly, unattended, and by using that kind of brute force, if you will, you can end up with a crystal structure if you're lucky. And typically that might be faster than trying to do it by NMR. The downside of that is that we need to keep in mind that the structural information that we're seeing is coming from a rigid form. It's a solid state, which is practically taking a picture, snapping time of how the protein might exist in solution. And something that it's important to keep in mind is that what we are seeing, the crystal structure, might not be exactly what happens in reality, because when the crystal is being formed, there are packing forces that might impact the shape or orientation of amino acids. So something to keep in mind when it comes down to X-ray. But the X-ray is more popular than NMR. But there are going to be cases where you might not have either of those, and then it's going to go to option number 3, which is create a homology model. If you are in luck, you might find, let's say, the crystal structure of a protein that is very similar, highly homologous to the one that you're working on, and you can use that as a template based on the amino acid sequence of your target of interest to build the model. But again, every time that you do something like that, you run into some unknown territory, because you're making assumptions every time that you start adding, let's say, a missing amino acid or changing amino acids. You don't know how is that going to, in reality, impact the results of the in silico model, because we don't know how close that's going to be to the real entity. But it's very it's well known that homology models work, so nothing to be afraid of. But we need to be aware of the limitations and assumptions that we're making. And when it comes to creating the 3D model, we pretty much go and we can grab a crystal structure, if let's assume that's available, and we pass it through a process. We're going to remove these extra entities that are not amino acids that we don't need. And it can be the water atoms, it can be ions, others. However, water atoms, water molecules can be kept if we know that they play a role in binding. So something to keep in mind. We also, once that we do that, we add hydrogen atoms to the amino acid chain. We fix missing amino acids if that is the case, and ultimately we have to calculate charges and apply those, because those are going to charges are going to play a role in calculations and modeling and docking. If we know where are we drugging the compound, or in this case, hopefully, let's say there's a small compound that's being co-crystallized, then we know where the binding site is, and we choose how big that is. Typically 6 angstroms, 7 angstroms, that's fine. You can go larger than that, but it's worth noting that the bigger the area that you are considering for exploring can not just only make the calculations larger, but you can start getting more potential binding modes or binding solutions that are not going to be relevant and accurate. And something very important nowadays is to make a decision as to are you using the protein alone in this case, or if it is available, in complex with other entities, because maybe in reality that's how that biological target of interest works. It has to form complex with other entities. Sometimes that's important. Sometimes you might get away with that by just removing the other ones and keep our target alone. But it's something to keep in mind. Now, when we come and look at the chemicals, we're going to have two sources. And in terms of chemicals, in this case, we're going to be creating virtual libraries. And I call actual libraries with actual compounds and theoretical compounds. And what do I mean by that? By actual compounds, I mean there are compounds that we know they exist. They're commercially available. They have been reported in the literature, or maybe that they exist because you have made them. Someone in the other lab has made it, or a partner has made them. So they're actual compounds. You're just creating, getting the 3D coordinates and using them, for your in silico studies. And if they're commercial, I mean, these entities here, I mean, they're a very good source of commercial available compound entities. And of course, you can go to the vendors directly too. And when we report that, we can get them from scientific publications or patents. And as I mentioned, whether you have them or someone that has those compounds. And then there are virtual libraries when we're using just theoretical compounds. And even here we have two kind of flavors. One is theoretical compounds that that they're possible to be made, and that's because you are basing new entities on a published or a synthetic route that you have developed, so that they can be made. And the others are the ones that you just find or design, but you don't know how can it be synthesized. So in that case, you got to be aware of that. Just because you are drawing a compound and you're making a particular variant, that doesn't mean that they're going to be synthesizable or stable or anything, or they're going to be even feasible to make them. Now, when we take these virtual libraries, we have to create the 3D coordinates of every single entity, every compound in it. And there's a little work that we need to do here to make sure that we have a high quality virtual library. These compounds are going to be eventually in solution because they're going to be tested in enzymatic assays and cell-based assays. Eventually, they're going to go in an in vivo system. So it's highly recommended that you protonate all the chemicals that you're considering, at a physiological pH of 7.4. You energy minimize the 3D coordinate, the compounds to get the 3D coordinates. That's something to get the coordinates themselves. And then let's remove the ones that we just simply think that we don't want. And that might be based on the nature of the compound. If he has reactive moieties, a microacceptor, could be epoxides, or the other type of functional groups that that you don't want to have in them. You can apply Payne's filters because some particular type of chemicals have known to just come and give you interfere with the biological assay, so you want to remove those if possible. If you're looking in druggability, you may want to draw a line or a threshold as to molecular weight, for instance, and also make a decision as to what type of moieties you simply don't want to deal with. If you say, I don't want nucleotides, I don't want sugars, I don't want peptodes, well, eliminate those from the pool, and that eventually leads you to a clean clean group of compounds for your virtual libraries that you can move ahead and use it for in silico studies. All right, so now we're gonna start looking into a little bit of strategy here, and we're gonna look into the strategy from the biological perspective and the chemistry perspective. When it comes down to the biological perspective, the biotarget, we mentioned, and now we start being a little more practical. How we're gonna get access to that compound? Can we buy it, or someone else has to make it for us? How easy it's going to be to express a biological target of interest, and how easy and complicated it is to isolate. That takes an impact in how much time it's going to take for you to get a hold of that. Something that is extremely important is the quality, because when you get the biological target, you start running your assays with it. You want to make sure that everything is reproducible. And if something changes from batch to batch, you're going to start seeing different results when you start changing batches, and that is just going to disrupt the whole process. So very critical, to pay attention to the quality of the biological target. And I mentioned, how we're going to try to drug that target. That the approach that we're going to use is going to be critical. Is it going to be going after a known active site, or we want to use an allosteric region to influence a conformational change in the target, or we might even want to stop a complex formation that activates the biological target, and we're looking into protein-protein disruption interactions. When we are designing compounds now on the chemistry side, we have to start looking into, how many different ways we can get to these compounds, how many synthetic routes, we have at our disposal. And then we have to look into how easy it is to carry out, that particular route. If it is going to be 4 steps, 3 steps, but one of them is going to require extremely high pressure, minus 200 degrees Celsius. I mean, something like that extreme. Then you say, well, clearly this is not something that I would like to consider. On the other hand, you can have a way to make a compound in 7 steps, but now you're looking into longer synthetic timelines. And as the third point shows, overall yields. Eventually, you need to make enough compound to do studies, especially if you need to start making more of it, to start advancing them and do more advanced tests. And eventually, making these compounds is directly going to be associated to the cost of the starting materials. If you find that one key building block it has a high cost, let's say $1,000 for I don't know 200 milligrams, well, that's something to consider unless you have a huge budget, to do something like that. When it comes to commercial compounds, then we're looking into, okay, are going to be available when we need to go and buy them? And again, how much is it going to cost us? Typically commercially available compounds for screening, they sell them in milligrams. So what's the price per milligram in this case for 5 or 10 milligrams? Timelines for the acquisition is important, because if you are dealing with a single vendor or several sources, one might say, oh yeah, I'll give you these compounds in 1 week, and maybe you have another one that is going to say it's going to take 6 months or 6 weeks. So that is going to start impacting on how quickly you can actually get a hold of these chemical entities to use them in the lab for testing. The purity of the compounds is very important, and in many cases, these compounds that are available, maybe 95% pure, 92, maybe higher. But there are occasions where the purity is low, 80 or lower, and in some cases, even the vendor says that this has been on the shelf for too long, and they don't guarantee the purity or any of that. So, got to keep that in mind. And ultimately, if something comes back active, you want to resynthesize it, because you want to confirm, this chemical entity, the chemical structure, and that purity is there. Now, when it comes down to the project itself, now we can start asking harder questions, and this is going to be now tied to what can we do, to move the project forward and get money for it. So this, I put here eight questions that I think can cover, you know, many things that are important to take into consideration. So why are you working or decide to work on a particular biological target? What is it? What is the opportunity there that you see? Why do you want to drug it in a particular way? Is it because you think you can do it better, or is it because it's going to be a new way of doing this and it's going to create intellectual property? Something to keep in mind. Um, what is the ultimate goal of the project? Are you looking into just advancing it enough for you to maybe get a grant, or do you want to really put something in the clinic? Getting clarification on that is very important. And what it is that you can do, what is under your control. If you have to do, let's say, steps 1, 2, 3, 4, 5, 6, and if you can control 1, 2, 3, now you know this is what you can do. You have everything that is needed, and you know where you stand. And now that takes us to the next question, which is the dependencies. Okay, so now that you know that you cannot do 4, 5, and 6, so how can you do that? You know, who's going to be able to carry out those things? You know, that are part of the research. And overall, you need to think about how far you think you can take this project. You have to be practical and say it can be based on funding. It can be based on the capabilities that you have. Like I said, also the dependencies that you have. So you need to know, if you start working on it right now, how far you can take it. And when start looking at eventually the money part, the funding part. So who else cares about the project? Who's going to say, oh, I get it. Oh, I see the value. I see your point. Yes, I want to give you money because I want you to continue doing this. So start figuring out, who else cares about this. And the other one, that's more a personal question, is, what do you want to get out of all of this? Once that all said and done, do you want fame? Do you want publications? Do you want patents? Do you want to put a camp in the clinic? What it is that you want to get out of it? I mean, you need to know your motivation in this case, which also start to the end goal ultimately. Okay, so let's go to the next slide. And when it comes down to the things that I do, for instance, I call what kind of engine you have, which is part of the what you control. In this case, when I work on a project, I can tackle the computational part and the medicinal chemistry input part, but I don't have labs. So here's a dependency where I said, okay, who can help me with this? And I work with a good partner, Supra Sciences. They make this synthesis happen for the compounds that the first process, the computational medchem, provide. And ultimately, this starts generating a lot of data. And I need to put that in some place where I can track it. I know where I can find it. I can mine it. And you might probably already know about the CDD, Collaborative Drug Discovery. Have a cloud solution. I can place there my information, my chemistry information, my biological information, raw plates, raw data. They calculate IC50s. A lot of good things. So that's the capabilities you need to know about what it is that you have. So now that we have a crystal structure, a biological target, of virtual libraries, start to get busy. The first thing is to actually validate, the 3D model. And typically, one way to do it is, if you know that there, a ligand, where a ligand binds, especially if it's coming from a crystal structure, you can extract the ligand, then redock it into the target. You can change here the number of times that you want to dock it. I think 80 to 150 is sufficient to get a good sense for how the docking model, the first pass is. Then you inspect the results, and then you see if you can get the results that you want, without any additional input. or if maybe you need to incorporate some pharmacophoric points there to help identify the binding modes that you're interested in. And this is going to be depending on what software you're using and the target. So now that we're mentioning what software to use, what kind of software we can have here, and we have free and commercial. The part of the free, if you look into publications, there's Doc, there's Autodoc, Fitted, Paradox, and there are many others. In fact, I think that you might be interested in checking out this website. You might already know of it, but it has a very good compilation of software, not only for docking, but a lot of processes that can be done in silico. And this publication also talks about what type of packages have been used, especially if they're free and open source. And on the commercial side, well, we have like our host today, Biosolvit. They have Leadit, or the next version, Caesar. We have also Biovia with SeedDocker, Schrodinger Glide, Mo, Gold, and Softmall. We have many others. And in my experience, some people sometimes ask me, so which one do you have to use? And I said, well, every software, whether it's free or commercial, has been created with particular algorithms, with particular assumptions, with their own specific way of calculating things, and that's how they differ. So in many cases, it's it wouldn't be unusual that you find that maybe 2 or 3 that you're using, they'll give you the solution that you're expecting. But sometimes maybe there's only one, and that eventually that's the one that you have to use. When you find that one of these work for you, just stick to it. It may not work for the next biological target, but for the biological target of interest at this point, just stick with that one that is giving you results. So in that regard, one that I like a lot, and I really wanted to mention it in today's presentation, not just because it's one from our host Biosolvit, it's because I really like it. It's Leadit, and now we have Caesar, which is the next generation. It's a very intuitive, straightforward way of doing your in silico docking studies. Caesar, the most recent next version generation here, even gives you some very nice visualizations of where you can position or are suggested to have particular functionalities. Let's say whether they're hydrogen bond acceptors of donors, or hydrophobic groups for hydrophobic interactions. So I want to show you here very quickly. I think I have 8 slides, but I'm gonna go quick here. What it is that you can see when you use Leadit. You have a nice interface. You go ahead and look into molecules to load up the protein, whether you have it in the hard drive, or if you just key in the code and go straight to the protein data bank server. You get it, then Leadit tells you what you have. And in this case, it's interesting that we have 2 chains displayed in blue and yellow, and the one in blue is actually showing the key chain where the inhibitor is. So we select chain A, changes in color to show that it's been selected, and then we proceed just to select where the molecule of interest is. We show it. May not show here very well, but amino acids around that, which is based on how big you want the pocket to be, are selected. In this case, 6.5 Armstrongs. It shows you what has been selected around the binding pocket. You can make changes in terms of keeping or removing water molecules or others. and then you're ready to set the model. And when that is done, you have a nice representation of the packet of the binding site, the ligand, and some electrostatics around that to give an indication as to what you expect to see for positive charges, negative charges, and the like. And here I want to mention something that is, in my perspective, is very important. Don't confuse simplicity with accuracy and the power of the engine. There was a lot of work that has been done here to make this look easy, to make it look simple. You can still do many of these things with other packages, or especially when there's not a very well developed GUI or process to do it. You can still do it with other applications, but at the end, making something look easy is actually not easy. So keep that in mind. So the docking is going to give us, with the model, is going to give candidates. We select which ones are the top based on a target affinity. And here's when we start looking into what kind of compounds that we have, the type of scaffolds, so we can start prioritizing, and then select which ones are going to be sent to test for activity. Typically, you want to get IC50s. In some cases, maybe because of resources, you want to start with percent inhibition at a particular concentration, and based on the inhibition percentage, you decide to get IC50s on those specifically. And ultimately, you want to de-risk everything, and for that, typically you want to synthesize these compounds and retest them to make sure that you can confirm the structure, confirm the biological activity, and to make sure that the biological activity is not coming from an impurity that was in the compound that you got. So now we are moving things forward. We have compounds of interest, and to advance them, to make sure that you're not putting everything in just one particular scaffold. If possible, you want to have 2 to 3. If you have one, that's still fine. But if you have 2 or 3 scaffolds, that gives you more variety. Or in case that one of them fails down the road, you still have another one that you can work on. How easy you can make analogs around that particular scaffold is going to be another parameter. You want to also change some key functionalities in the chemicals, and this is because we want to confirm activity, particularly killing activity. Then oh, this functional group is essential. I'm going to keep it, and it gives you a sense for how to start developing a structure-activity relationship. With this new information, you can create new virtual libraries that are going to be more focused, and you can redoc them to start getting some guideline moving forward. And typically, it's good to use one of the compounds that came back as good hits from the docking exercise. Using is calculated binding affinity as the threshold. Anything that is going to be more potent than that, based on that particular engine, is going to give you an indication that in theory should be, if not at least at the same potency of that compound, or even more potent. But I mentioned earlier on that you might be dealing with a biological target that for which there are other biological entities that are very similar to that. So we can call those anti-targets. Those are the targets that you don't want to hit, because that can lead to unwanted toxicities. So the process is practically the same. You identify them, you create the 3D models, you validate the models, and now once that you dock, let's say your compounds against the target of interest, now you can come and take the top ones and dock those and against the anti-targets. And you can start eliminating this way the ones that are going to be hitting both targets. Those are going to be more promiscuous versus the one that hit only the target of interest. Now, when we are not doing tests in the lab, you want to eliminate very quickly those that are going to give you trouble. And and the way that I've done that in the past, many of you might disagree here, is doing cell-based assay. If Because if you see an effect, a desired effect in a cell, then you know that the compound is entering the cell, therefore it's cell permeable. If it's creating a particular effect, then you know that it's doing, I mean, it's creating the phenotype that you want. And at the same time, you can confirm that the compound is hitting the target that you're going after. So you want to confirm target engagement. From a chemistry perspective, well, the compounds have to be soluble in water eventually. So you want to eliminate those that are going to be soluble versus the ones that are going to be just, bricks that they're not going to go into solution. So they're going to be problem makers. So you can eliminate them both, those early on. And eventually, just to get a hint of, ADME properties and how these compounds might behave, or what kind of profile they might have, let's say from a pharmacokinetics approach, for instance, I like microsomal stability. Why? Well, because it's a very, very easy test. You mix it with, let's say, microsomes and your compound of interest for half hour, and then you test. How much compound is left after 30 minutes. If you find that your compound it still remains, let's say 90% of it still remains, then that's a compound that is going to be stable. It's not going to be metabolized in the liver. But if you find that in 5 minutes is completely or practically all is gone, then you know that even if it enters the cell, even if it can be water soluble, once that it goes into an in vivo system, it's just going to be eliminated. So you might want to use that early on as a filter to remove potential compounds that are going to fail anyways. And the money. I want to start here very quickly with the mindset about money. And that is, many of us can make a very, can fall into a trap, and that is, treating money and time in a different way. And money and time, they're not infinite resources. Money goes without saying, but people say, oh, I can always do the experiment tomorrow. Oh, I can do this in a month. Well, that eventually is going to have impact, not on the project, and eventually even in funding. One thing that happens with money is that everybody wants to save, and that's fine. We all want to save money, but if you focus too much on buying everything, that is the cheapest, to save as much as you want, that is not cost efficiency, because you're going to now be, for lack of better words, wasting a lot of time, because you want to save $2-3 here or there or something. So sometimes, yes, we want to buy, the cheapest possible, but keeping time into consideration, just get a couple of good options and just make quick decisions. So you can, this is going to help moving the project further. Time kills funding. Why? Because even if you are not doing anything, you are, it's costing you money. The labs are running. You're paying the people. You are paying for the instrumentation, the maintenance. So you are still paying for things, even though things are not happening. So yeah, time kills funding, and time kills project. Why? Because whatever was of high impact today might not be of high impact in 2 years. There's going to be competition that is going to pop up. There's going to be new discoveries, and eventually what was novel today in 2 years might be obvious. So you need to move quickly, because no funding equals no project, and eventually your group might not even exist. So I'm going to give you some places to consider to go and look into funding for industry, not-for-profit, and academia. For industry, there's different type of investors. Can be friends, can be angels, venture capitalists. You can also get grants, whether coming from the government or from foundations. Something that is very important to know is that many large pharmaceutical companies now, they have their own investment branches. So so they don't do just drug discovery; they also invest. So it's a good way to look for opportunities there. There are companies that they are scouting for opportunities, whether they themselves invest in their venture firms that are well known. This is just an example of what it's out there. The list is long. You can find many that out there. Government, if you're in industry, sbir.gov is a very good source to go and get funding by submitting ideas. Department of Defense, if that's something that uh, your particular application can be used in, and foundations, the Bill Melinda Gates like Foundation, multiple myeloma. If you're in cancer, that's one example of the type of foundations that you can look for funding. Non-for-profits, typically non-for-profits get funding by either gifts, donations, grants. In many cases, they have their own products and services, and they actually operate like a regular company, except that being non-for-profit, they have to break even at the end. They cannot make a profit. So in this case, there are foundations out there that can non-for-profits go to, government as well, and some sites and places, endowments that you can find. And I believe Marcus said that we're going to make this video available, so you can come back and take a look at these websites and the names, so you can search them yourself. And last but not least is universities. Universities get money from grants, endowments. Something that is actually coming up more now is entrepreneurship programs. So that's something worth for you to look into a particular university, see what they have. And of course, foundations. You have government, NIH, everybody knows about them. In terms of what universities are doing, we have, for instance, Duke University has a department called Research Funding. Columbia University has a department called Finding Funding. So look what what your school has and what university has, and you might be surprised to find some opportunities there. There's also this one. If you are international, you can go to Wikipedia and look for funding of science. You're going to find very good sites that give you good pointers if you live in different countries. What opportunities they might have. It's not comprehensive, but it's a very good place to start. So at this point, I'm going to ask you something here eventually after this, but these are new skills that I found are very helpful to you, as I mentioned, in life, whether in the lab or outside of the lab. And that is communication. That's important when it comes down to presenting to others. Finance, when you know a little, when you know more about money, how interest and the type of different accounts that you can have, how you can make a little bit of investments, etc. And sales. That's another part of business. So, and is there and what I find very interesting is that this happens. I mean, these areas are very important, but at least when I was in school, they didn't teach them in the chemistry department. If you had any time, you might go and not go and look into other departments, but they're not part of the curriculum. So this is something that was learned once that I got out of school. So so the one question that I have here, which is we're going to put in a poll right now, is which of these 3 skills you like the least? This is the one that you say, oh my gosh, I like these, but this one I not feel so cozy about. So Marcus, can you help us with the poll?
Yeah, absolutely. Thanks very much for this point. Yeah, I put up the poll and people are actually clicking rapidly already. Yeah, it would be interesting to know what you rather run away of, what you dislike most, what you really don't like. And there is a clear winner coming out here, a clear dislike winner. So I'm going to close in 3, 2, 1. Now, excuse me, I'm sorry. Convey the results. Sales. Selling stuff. That's what people don't like. Very.
Well, that's it's good to know, and I have to say, to some degree, I was expecting that, because no one, a lot of people don't like sales. They think that it's cheesy, that it is something beneath them, it's something that is not appealing. But I just wanted to mention in less than a minute that of all of the skills, probably sales is the most important one. And it's not because you need to be manipulative, or you need to be like a car salesman, pushy. There's a lot of psychology that goes behind sales. There's techniques that are not pushy, that can help you talk to potential people and still do a sale. But sales is not about go sell a product. Sales isn't even selling yourself, or selling your program, for instance, as an investment, as in, hey, when you apply for a grant, you are selling yourself, you are selling your program, you're selling it to the review panel. Hey, this is what I have, this is what I think you need to give me money. Or if you happen to have face-to-face, like a job interview, you are selling yourself to the potential university, or potential place that is hiring. So sales is more broad, and eventually, again, that helps you not only personally. I mean, that's something that I had to learn to look for, to create my business and make it sustainable. But if you are in research, you need to find, like we were talking about, money, and you need to sell your project, you need to sell your target, you need to sell your approach, you need to sell the impact of this. So keep that in mind. And when it comes down to sales, might give you some enough interest to look into that a little further. But I just wanted to put a little context when it comes down to sales. And lastly, I want to give, to wrap it up, a quick take-home message, and that is, don't spend a lot of time trying to get any of the process perfect. Just get it to work, and you need to get moving. We said that time kills projects, so that's what you want to do. Just move quick. I mean, do good work, but again, don't try to go for perfection. If you're gonna eliminate compounds, eliminate them fast, quickly, and as early as you can. People might say, "Oh, but I'm gonna end up with 2 compounds only." Yes, but those 2 compounds, are now the points that have better quality, they have better properties, a better profile, and that's what you want anyways. So, I mean, eliminate the noise. And lastly, don't get paralyzed by trying to save money and taking a lot of time making decisions. Just go ahead, make a quick decision and get on going. So at this point, went over for a few minutes here. I want to thank you, everybody, for listening. Thank you for Solver for inviting me and giving me the chance to give you this seminar. Feel free to contact me if you have any questions, or you would like to have a conversation with me. Maybe you can sell me your idea, or you want to talk about it. I mean, that'll be fine. And again, thank you very much, and very happy to take your questions.